When a neuron receives one of these volleys I showed you, the volley carries surprise because all the component spikes, which could be scattered all over time, are all concentrated in a narrow time interval around the volley. But there's another source also of the surprise to the volley, and that is that the component spikes, which could all come from various cell types, all actually come from the same biological cell pool. For instance, if the volley comes from orientation selective cells, then all the components come from cells with the same preferred orientation. Neurons in the brain carry their signatures in the form of specific cell adhesion molecules. These molecules determine the way the cells make contact, and therefore, to a large extent, they determine the functional properties of the cell. Now, at the output end of the cells, these same molecules are around at every synapse. Therefore, the next cell is able to tell, at least is in a position to tell, at every synapse the properties of the presynaptic cell. So in a sense, cells have synapse pools in accordance with the presynaptic cell type of the synapses. The argument that the cells actually make use of this information from their synapses is that if, for instance, orientation selective cells could not pass on their orientation preference to any of the cells they make contact with, then that orientation preference information would be lost. Now, it is very likely that the orientation selective cell as a functional cell type is only the tip of the iceberg, and that even in the visual cortex there are plenty of other cell types of various functions. Of these, I would like to discuss one whose existence has not been demonstrated, but which is so similar to certain known cells that I will assume that they exist. These cells, which I call direction-coded cells, seem to be indispensable to shape processing. They have long horizontal axons. Cell assemblies of such cells send their axons into narrow sectors of the visual projection, like this. In that way, they are selective to the direction in which their effective field is located relative to their receptive field. The effect of volleys sent out from such cell groups is seen by considering the signals reaching a cell located within the field of their axonal arborization. One has to imagine that the cell in question is also reached by other similar groups like this. Now, let us say that on one occasion the marked cell receives firing like this. Here I separated the spike trains coming to different synapse pools on the cell and indicated the pools with small drawings at the left. What makes the spike trains special in this case is that the middle set of tracks shows a series of volleys and the other tracks only show random firing. This means that if the neuron in question can read the labels on its input terminals, it is in a position to know which direction the volleys come from. This has far-reaching consequences, as I will show in the next videos.